So we are continuing with the part from the genetics, that is principles of inheritance and variation. Now I am going to start with the topic, that is chromosomal theory of inheritance. This theory was given by Suter and Boveri in the year 1902, but the experimental verification was done by T.H. Morgan. He did his experiments on Drosophila melanogaster. Another name for Drosophila melanogaster is fruit fly. And he found this chromosomal determination in various organisms. We are going to discuss this topic, that is linkage. Linkage is tendency of two genes to be inherited together on a particular chromosome. That is known as linkage. Recombination is the combination which is formed depending upon the distance or the linkage. So they both are related to each other more, and they are also related to the distance. More the distance, less is the linkage. More is the distance, more is the recombination. That means more recombinants are formed if the distance is more. But if the distance is more, the linkage will be less. Okay? So I've just now given you a relationship between recombination and linkage, distance and linkage. Okay? Now coming to determination of sex in humans. We have two types of chromosomes, autosomes and sex chromosome. Autosomes are the chromosomes which are present in the somatic cells. Sex chromosome, in the case of male it is X and Y, in the case of female it is XX. So this is for human. Males are heterogametic, females are homogametic. Even in the case of most of the insects, the male is heterogametic. That means they have two different types of chromosome. In case of grasshopper, we have, in the case of grasshopper, they have autosomes plus X0 type. In case of male, we have X and Y, they have X0. That means there is no chromosome along with the X. And in the case of female, it is same as in the case of human female, two autosomes and two sex chromosomes, as they always occur in pair. In case of birds and reptiles, it is different. In autosomes, female is heterogametic, male is homogametic. That is why we are using this ZW and ZZ. Why we are taking this ZZ or ZW? Because in case of human, the female is homogametic and male is heterogametic. But in case of birds and reptiles, the female is heterogametic and male is homogametic. In order to reduce the confusion, we are using these ZW and ZZ. In moths and butterfly, they have is AA plus Z0. That means female is heterogametic with a single chromosome. And in case of autosomes, it is AA. In case of male, ZZ and AA. But there is a special case in case of honeybees. Honeybees, they show haploid-diploid relationship. If it is haploid, if the egg is reproducing without fertilization, that is parthenogenetically, it will lead to formation of male organism. And if it is produced from a diploid egg or fertilized egg, then it will produce the female. The female is produced from diploid egg, that is fertilized egg, after the process of fertilization, and males they are produced without fertilization by the process of parthenogenesis. So let us start with this topic, linkage. Linkage was first studied by Suter and Boveri in Chromosomal Theory of Inheritance. Later on, Morgan in 1910 did experiments on fruit fly, that is Drosophila melanogaster. Okay, he found that linkage is of two types. It may be complete linkage or incomplete linkage. He studied that the red eye colored and the grey body, these two genes, they were inherited together in the next generation. So complete linkage, it is the tendency of the genes to be inherited in the next generation even after this crossing over. It is maybe because of the distance present between the two genes is very less that they are always linked together. Incomplete linkage, that means it is not coming in as parental type in the next generation. It may occur because of this process of crossing over. What is crossing over? It is the process where the genetic material between two homologous chromosomes, they are exchanged during the process of meiosis. And it occurs at Pachytene stage because of formation of this chiasmata. Chiasmata is the result of this crossover. It is not because of this chiasmata formation point of crossover, that linkage occurs. Okay. Now coming to very important topic, what are mutagens? Mutagens are the agent which causes mutation. It can be of two types, it is physical or chemical. In the physical, we can say that it is because of the radiations. Radiations may be ionizing or non-ionizing. Ionizing, that means penetrating. X-rays or cobalt rays. Okay. X-rays helps in breaking the T double bond T. That is TT dimer. Okay. Non-ionizing rays, example is UV rays and gamma rays. They are non-ionizing rays. Even the temperature helps in denaturation. It has a mutagen also. Even we can say the sound is considered as physical mutagen. In case of chemicals, they can alter the base sequence. For example, all the nitrogenous compounds. For example, nitrous acid. It is an example which can change the base sequence. It may add or delete the base sequence in case of DNA. For example, acridine dyes. Okay. Now abnormal, they may show abnormality. They may enter or they may join in with the DNA chain, for example, 5-chromouracil. So with this, we are done with the genetics first chapter. I hope it is clear with you. If you have any kind of query, just let me know. Thank you. Good luck and God bless.